Friday, the best day of the week. And it's only going to be by the grades that are going to be posted later today, right? Hopefully it's not going to be too bad. Okay, so let's just keep trucking along. Still quite a bit of stuff to cover. What's going to happen is, especially next week, you'll notice that I have posted slides and I don't show them in class. So again, the rule for that is if I don't show it in class, it's not on the exam. That's it. Okay, so don't worry about it. Because of all the sidetracking that I do, I always tend to get behind early on. But I'm so happy about the invention of PowerPoint slides. I can really start picking it up quickly too. So, and as you know, you can always slow me down, right? How do you slow me down? Yeah, answer questions, or don't answer questions. Just let me be quiet here for the whole freaking hour, okay? Okay, so let's recap really quickly yesterday. So because I really want to finish the sex determination part. This should have been done in one lecture, but clearly this is lecture 3. So we talked about chromosomal sex determination. We finished talking about primary sex determination. And we started talking about secondary sex determination, okay? And secondary or phenotypic sex determination really involved having a careful look at Alfred Joseph's experiments, okay? So Alfred Joseph, his experiments are important. And I don't care if you, actually, I expect you to write it down right now. There will be, people are starting to throw stuff at me already. There will be, you're fine. There will be at least one, probably 2 questions related to Alfred Joseph's experiment on the exam. Very good. Everybody writing that down. Crap, this freaking Alfred Joseph, he's killing me. Okay. With Alfred Joseph's experiments, we really only focused on the internal tract, right? So what we're going to start with today is we're going to focus on the external genitalia, okay? So when we start talking about the external genitalia, so far we already talked about several things that are bipotential, okay? Wonder if you guys can name 4 things that are bipotential that we've talked about so far. Which one? What? Gonads is one, right? Bipotential gonads. Doesn't get any easier. Mesonephros. I love it, right? Because the mesonephros is bipotential because whether you're XX or XY, initially it houses 2 ducts, the Wolffian duct and the Mullerian duct. And it really depends how the gonads differentiate whether or not the Wolffian duct stays around or the Mullerian duct stays around. Okay. What's the third thing? Germ cells. I love it. Yes, germ cells in a way are bipotential. Especially considering, regardless of whether they're XX or XY, it really depends on the environment that they are in on how they're going to develop, either as spermatogonia or oogonia. Right? It was really what happened to retinoic acid, if it's going to be metabolized or not, but that was all the somatic cells surrounding it that caused that. And then finally, the bipotential external genitalia. Again, it could go one of 2 ways. Okay? So when we started talking about Alfred Joseph's experiments, what did we learn from that? What really is he famous for? Besides being maybe a freak that does weird things with rabbits and rats. Very good. The effects of testosterone, or really the role of testosterone on the internal reproductive structures, right? So where did the testosterone come from? From the fetal testis. And can we even be more specific than that? Where in the fetal testis? This is kind of the tutoring session we're doing again. Leydig cells, right? Leydig cells. Leydig cells make that fetal testosterone, gets into the bloodstream, goes towards the Wolffian duct, and the Wolffian duct survives and actually develops into the epididymis, the vas deferens, and the seminal vesicles. So again, you can write this down too. I think I told you that yesterday too as well. I expect you to know for the exam the structures that come from the Wolffian duct. I expect you to know the structures that come from the Mullerian duct. Okay. So testosterone was very important, but you know, Alfred Joseph was not really the guy that discovered testosterone. People already knew that testosterone existed. And in fact, even, I'm pretty sure at least 100 years before him, or maybe even 200 years before him, people were already grinding up testes And then if you do that in a tube in some water, they would take the liquidy part, the watery part, and inject themselves with that. Because they really felt that that helped with general performance and made you feel really good. Do we remember what testosterone is? A hormone, but a steroid hormone, right? So if you do something like that, I know we all love chemistry, where would that hormone be? Would it be in the watery fraction or not? No. So basically, we had lots of people that were injecting themselves with, I have no idea what, but some watery stuff from a testis. Okay? Whether or not that made them feel very good or happy, we'll call that the placebo effect. Okay? In some cases, there's definitely a placebo effect with lots of things. People just feeling better just knowing that they took something. Okay? Anyway, Alfred Jost was not famous for testosterone. What was he famous for? Discovering what? AMH, anti-Müllerian hormone. So I want you to remember again that AMH, or anti-Müllerian hormone, does not come from Leydig cells, but comes from Sertoli cells, and also is not a steroid hormone, but a peptide hormone. Okay? So this peptide hormone also gets released into the bloodstream, and this peptide hormone binds receptors on the Müllerian duct, causing the Müllerian duct cells, there's just a duct with epithelial cells, and it causes apoptosis in these epithelial cells. So the cells just die and the Müllerian duct disappears. Okay? So again, especially as we continue talking about reproductive physiology and male and female, keep track of what is a steroid hormone and what is a peptide hormone, okay? Because that will dictate where we can find receptors, on the plasma membrane or inside the cell, things like that, okay? So AMH is important. Okay, I think yesterday we were close to being about here, the external genitalia, bipotential gonads, okay? So we talked about the internal stuff, okay? Clearly the fetal testis secretes testosterone, AMH. The ovary does not secrete any AMH, obviously. Can you imagine if the ovary secreted AMH, what would happen? Yeah, you would, the female would not have a uterus, no fallopian tube, no cervix, and no upper portion of the vagina. Okay? So the ovary does not secrete AMH. Does the ovary secrete testosterone? Yes or no? Okay, let's hold on to that question. Okay? Let's finish this stuff. Okay, so the fourth thing that is bipotential, external genitalia. And again, you guys already knew this because couples won't find out whether it's a boy or girl via ultrasound until the 20th week. Okay, because that's when this is, not to the extent of this, but that's when you really could tell the difference between a boy or a girl. Okay, so bipotential is boxed in red. We have the urethral folds, or the urethral groove, that fuses in the male, and it doesn't completely fuse in the female. We have the labial scrotal swelling. That one enlarges and fuses and develops into a scrotum in males. Does not happen so in females. Okay? And then finally, the genital tubercle that elongates, develops into the penis in the male, and kind of regresses and is the clitoris in the female. Okay? So again, this is driven by hormones. Secondary sex determination or phenotypic sex determination is driven by hormones. What is highlighted or boxed in red, that tissue right here has two things that you really need to know. Okay? So the one thing is 5-alpha reductase. Okay? And the second thing is androgen receptor. Okay. Whether you're XX or XY, the cells that make up that tissue, bipotential external genitalia, have that. They have a receptor for androgens, and they have an enzyme. And this enzyme takes testosterone, or T, and converts it into dihydrotestosterone, or DHT. Okay. Testosterone is an androgen. Dihydrotestosterone is an androgen. The only difference here is that dihydrotestosterone is way more potent. Okay? It has a much higher affinity for the receptor, And so you would really need a lot less in order to cause a similar response. Okay? So if testosterone does something to cells, and let's say making some odd number up, 10 nanomolar, then dihydrotestosterone could do the same thing at maybe 0.1 nanomolar. Does that make sense? Higher affinity, same kind of a chemical. It binds the same receptor, but it's more potent, it's more powerful. Okay? So dihydrotestosterone. So all that stuff is present right here. Now what happens during normal development, and obviously this is adult male reproductive tract, but it kind of highlights the structures that are dependent on DHT versus testosterone. So what happens is that testosterone, as it is released into the bloodstream, is converted to 5-alpha reductase, by 5-alpha reductase into dihydrotestosterone, binds the androgen receptor, and then causes this pathway to occur. So we're pushing the bipotential gonad towards development of a scrotum and a penis. Okay? Does that make sense? You need testosterone. In this tissue, it's converted into dihydrotestosterone, causes development along this pathway. Now, it may make it sound that development along the female pathway is some kind of a passive thing that will just kind of happen by itself. That is really not true. The only thing is that this part, the female pathway, does not rely on hormones. It relies on genetic factors that are not regulated by hormones. Okay? So this is not some kind of default pathway. Stuff still needs to happen. Okay? Okay, so what we're going to do now is we're going to look at a couple of clinical cases that will really kind of help test your knowledge about how well you understand Alfred Joseph's experiments. Okay, so we did all this stuff with Alfred Joseph's experiments focusing on the internal tracts. Now we're going to use some clinical cases to kind of figure out how does this apply to external genitalia. Okay, now I have to apologize, they're kind of out of order, but I'll help you go through this. The first one is this. Situation, okay? So this is an individual that is XY, okay, and has complete androgen insensitivity syndrome. What complete androgen insensitivity syndrome means is that there is a mutation in the androgen receptor, okay? So there is an androgen receptor, but it is mutated, okay? It doesn't work, it doesn't function, okay? Is there anything wrong with the chromosomes in this individual? No, right? The total number is 46 chromosomes per cell. And the sex chromosome, there's one X and one Y. There's not an odd number of sex chromosomes or anything like that. So chromosomally speaking, in terms of numbers, there's nothing wrong. Okay? What does this individual look like? Yes, I would hope that that is going to be the easiest question that I'm going to be asking from here on, okay? Clearly this is a female, okay? Now before we go back to this case, okay, I want to focus on a different one that is not XY related, but XX related, okay? So this is a situation where a baby is born with clearly ambiguous genitalia. Okay, so if you're a doctor in the delivery room, the baby is born and it looks like this, what are you going to tell the parents? Because remember, they only care about one question, right? They're going to assume it's healthy. They're going to assume it has one head and two arms and two legs. So how are we going to answer this question? Sorry? No words, just quiet. Now you have mom there. She's still kind of half recovered, half sedated. She's been swearing her head off for the past day almost. Dad is getting pretty freaked out. They want to know something. You understand the problem here? Okay, because you cannot really, what are we going to say? There is no sex. Sorry, it's neither a boy or a girl. Trust me, that's not what they want to hear. Okay. So there is something kind of more of a generic term now that is called disorders of sex development. Now clearly that's also something that parents don't want to hear, okay? And basically what the doctor is forced to deal with or tell the parents is that we cannot assign a gender to your baby, okay? Which clearly now we're going down a path that does not only involve the doctor having to make decisions, it's going to involve the doctor and the parents and psychologists and counselors to get together, and this could go on for years and years and years, okay? In this case, the first thing that the doctor needs to know, okay, is this baby XX or XY? Now, I put it up top here, but if you ignore that, if you just look at this, can you tell if it's XX or XY? Because there's two possibilities here, right? It's either an XX baby that it became masculinized, or it is an XY baby that is under-masculinized, okay? So the first thing that happens is they can either take some blood samples and then they do a karyotype analysis. And basically that just means we collect some cells and we look at the chromosomes that are there. Okay? We count the chromosomes, make sure there's a proper number. So in this case there's 46 chromosomes. Okay? And they also find no Y chromosome but 2 X chromosomes. So in this case we're dealing with a baby that is genetically, chromosomally XX. Okay? Okay, so now what can you tell me about the rest of this baby? Okay, so this baby is XX, and eventually what it is diagnosed with is something called congenital adrenal hyperplasia. Okay, hyperplasia or congenital adrenal hyperplasia just really means that the adrenal gland is really active. Okay, has Dr. Walrond ever mentioned the adrenal glands? Was that a couple of weeks ago, long time ago, way too long ago? What do we remember from the adrenal glands? Where are they, besides the obvious answer in our body? Top of the kidneys, okay, top of the kidneys. Anything you could tell me about the adrenal glands? Is there anything odd about it? Steroids, it produce steroids. So remember again, adrenal gland, right? There was a cortex on the outside, a medulla in the inside. Okay, since you guys have a comp exam, so my BMS 360 class do not have a comp exams, but you guys are way smarter. You can deal and handle a comp exam. So what does that medulla secrete? Ooh, I can see sheer anxiety popping up now. What the hell? Do I have to study all this crap? Do we remember? No, not that one. Epinephrine? Do we remember epinephrine at all? Kind of. Okay, so that comes from medulla, norepinephrine, epinephrine. And then the cortex. And I already heard one hormone secreted by the cortex because you guys are all experts in renal physiology. Even though the grade tonight may not reflect that, you are, okay? So the cortex, aldosterone. What else? What else does the adrenal cortex secrete? Can you think of a hormone that regulates blood glucose levels? Yeah, that's insulin, but we know that insulin doesn't come from the adrenal gland, right? Pancreas? Sorry? Glucagon, also from the pancreas. Yeah, also regulates blood glucose. There's another one. Epinephrine does, but we already covered epinephrine. We're running out of options. Sorry? No. It actually even starts with glucocorticoids, cortisol. Cortisol, okay? Cortisol regulates glucose levels, glucocorticoids. Does it have anything to do with reproduction? No, absolutely not. I'm wasting time. So what is the third hormone, okay? The third hormone is what? That's the one that I really care about. Well, in reproductive physiology, it's important. Yes, testosterone, yes, testosterone. So in addition to getting, again, a little bit of a tutoring session for your final, I wanted to get to testosterone, okay? So if we have congenital adrenal hyperplasia and the adrenal gland is overactive, that means the adrenal gland is making a lot of testosterone, okay? And usually that's due to mutations that prevent making glucocorticoids or cortisol or aldosterone. And because those pathways are blocked now, everything gets shunted towards making testosterone. So basically, the mom could either be the mom or the fetus, it doesn't matter. They make a lot of testosterone, okay? So how does that explain this phenotype? So this baby started to develop and sees a lot more testosterone than normal. How does that explain this? Yeah.
The gonadal sex was determined already, and then too much testosterone is produced?
Yeah, so there's definitely too much testosterone being produced. And think about starting with a bipotential gonad, and there's lots of testosterone. So what's happening now in this XX baby?
Interrupting the development of female.
Not so much interrupting development of the female, but it is promoting development of the male, right? What did the external genitalia have? 5-alpha reductase, causing it to make DHT, an androgen receptor. It doesn't matter where the testosterone comes from, okay? In this case, if mom has a lot of testosterone because her adrenal glands are overactive, or not mom, but the baby makes extra testosterone from the adrenal glands because of the adrenal glands having issues, that testosterone gets into the bloodstream, it goes towards the bipotential gonad, it gets converted to dihydrotestosterone, binds the androgen receptor, and we start developing male external genitalia. Now, the reason why this is not looking like a completely normal male baby boy is because it has to do with how much testosterone was there. So there was definitely enough to start initial development towards the male pathway. Does that make sense? Okay. So what kind of gonads does this individual have? Who says ovaries and who says testes? Do ovaries first. Who says testes? Okay. So the answer is ovaries. Okay. And the reason for this is, in order to make a testis, do we need any hormones? In order to make any ovaries, do we need to make any? Do we require any hormones? No. In both cases, we do not need hormones to make gonads. It's in fact the other way around. Okay? The gonads make hormones, okay? And all the rest requires hormones, okay? But we do not need any hormones to make testes or ovaries, okay? So what do you think internally? Internal reproductive tract. So think about the Wolffian duct and the Mullerian duct. What is present? Absolutely right. Chromosomes, right? If it was XY, we have a Y chromosome, SRY, testis. If it is XX, we have RSP1, Win4 beta-catenin, we make ovaries. That's it. No hormones, just genetics. That's it. Okay. What about internally? Does this individual have a uterus, cervix, fallopian tube? Yeah, right? As long as there's no AMH, that stuff will be there. AMH only comes from testes. Okay. What about Wolffian duct? Yeah, there's testosterone, right? And as long as there's testosterone, that's Alfred Joseph's experiments, right? You add testosterone, there will be a Wolffian duct, okay? So clearly, this one is very difficult because this will require either a lot of surgeries, decisions that need to be made, gender reassignment or not, because if we start with just, well, I'm going to go whatever my chromosomes are, that means that chromosome-wise, it's a female. Now, on top of that, internally, there are ovaries. But after that, it's going to require some extensive surgeries if we really want to continue down the female path, okay, both internally with the tracts and externally as well, okay? When is this diagnosed? Well, when the baby's born. Okay. Now let's go back to the other one. This is the same, and I have to apologize. I don't know why I keep flip-flopping back and forth like this, but again, this is just a different individual, but the same syndrome in terms of complete androgen insensitivity syndrome. Okay. Again, I wrote down mutation in androgen receptor, meaning it cannot respond to testosterone. Okay. The androgen receptor is not working. Okay, so let's quickly do this. What is the chromosomal sex? It's XY, right? Yeah, XY. What kind of gonads does this individual have? Use the same train of thought as that we did with the previous one. Testes. Can we see the testes? No. Well, not on this picture. Undescended. Undescended testes. Okay, but yes, testes. Testes would have developed. Again, the easiest question ever. Phenotypic sex, female. Okay, so what I want you to do now, because I know you guys are going to be bored out of your minds after you leave this class, and you have nothing to do this weekend anyway, right? So a little homework assignment for yourself to see if you really understand Alfred Joseph's experiments. So my question is, and I'll answer it on Monday, is internally, what is present in this individual? Okay, so what is present in the individual related to Wolffian duct derivatives and Müllerian duct derivatives? That's it. Okay, so try it out. Obviously, I don't care whether you do this or not, but for yourself, it's a good exercise if you really feel like, you know what, I've got Alfred Jost down. Okay, I can handle this stuff. Because what I'll do on the exam, again, I'll take a variant of this. Okay, so it's really important that you kind of know what's going on here. Any questions about this stuff? Pretty straightforward? Yes?
A person with that syndrome, would they be infertile?
Would they be infertile? Yes, they will. Okay, so because I don't want to go towards whatever, but regardless from that, so what we'll talk about in a minute is, we're going to go into male reproductive physiology, is the fact that testes are weird, right? They are very weird because it's the one organ that's not inside our body cavity, right? Yeah. So if you keep the testes inside the body cavity, it doesn't work, it doesn't function, and you're more likely or not to develop testicular cancer and be completely infertile. So I'm glad that you brought up this question, because my question back to you, when would this individual have figured out there was a problem? Yeah, puberty. I mean, at the time of birth, this looked like a healthy, young baby girl, okay? But by the time puberty rolls around, she would have noticed something was not happening. And so probably the first thing, obviously, this woman is way past puberty and well into, you know, not well into, but an adult, okay? They would have already removed her gonad. So even I put testes there, undescended testes, that picture, that lady has no gonads, okay? They would have been removed, okay? Any other questions? Okay, let's do the easy stuff first. Male reproductive physiology, okay? Basically what's going to happen now is hormones, and there's going to be different cell types, and what I would strongly recommend and suggest, anytime we mention a hormone, ask yourself, where does it come from, where does it go to? And by the time we ask where does it go to, that means the receptor for that hormone is there, okay? Second question is, you mentioned hormone, is it a steroid hormone or a peptide hormone? That's it. So this is what I took from the ebook that you hopefully, possibly have access to, the same lecture objectives, okay? So this first slide is kind of a shitty slide, but it does have lots of colors. The only point of this slide is to kind of remind you again about endocrine systems, right? And if you talk very broadly about endocrine systems, it involves the hypothalamus, the pituitary, and some kind of target organ, okay? In this case, the target organ is the gonad, okay? So in terms of the endocrine system as it pertains to the reproductive physiology, we're talking about the hypothalamus, the pituitary, and testes or ovary, okay? Now, what's kind of crappy about this slide is that it tried to combine male and female together, and it really gets awkward here in this blue box. So just kind of ignore that part. If we start on top in the brain, okay, there is some environmental or internal stimulus, okay, that will act on the hypothalamus and specifically on neurons that secrete GnRH, gonadotropin-releasing hormone. So all the peptide hormones coming from the hypothalamus are releasing hormones. Okay, so GnRH, a peptide hormone, is released into the portal vessels that are right here in the infundibulum, and so that peptide hormone goes to the anterior pituitary gland. Dr. Warren talked about the anterior pituitary gland. There are different types of cells that are around. They all secrete different types of peptide hormones, trophic hormones. Okay, The ones that are important for reproductive physiology are called gonadotropes. Okay? Tropes means, or from trophic hormone, means nourishing. Okay? Gonado because they act on the gonads. Okay? Now the two that we need to know are LH and FSH, luteinizing hormone and follicle stimulating hormone. Okay? So LH and FSH are both also peptide hormones. They get released into the bloodstream and they travel towards the gonads. Okay? Now, yesterday we talked about different cell populations that are present in bipotential gonads. One of them was called somatic support cells. Okay? They support germ cells. Okay? FSH acts on that cell population in the adult. That means FSH acts on Sertoli cells and granulosa cells. Okay? LH was the one that stimulates the endocrine cells or the steroidogenic cells. So in the testes, that means Leydig cells. In the ovary, that means theca cells. Okay? Now, ultimately, this leads to negative feedback. Back to the anterior pituitary gland and back all the way to the hypothalamus. Okay? Does that make sense? So fairly straightforward endocrine system, hypothalamus, pituitary, gonad, and negative feedback. And these are the hormones that we're really going to be talking about. GnRH, LH and FSH, and feedback by testosterone and another hormone we'll talk about later. Okay? What gets awkward here is that LH, females only gamete production. This just means that by the time we talk about female reproductive physiology, LH does not only bind theca cells, but LH at some point, a very specific point during the reproductive cycle, acts on the egg. Okay? That's really it. So LH does not act on sperm directly, it acts on Leydig cells. Okay? Now, because we love our steroids and our hormones, why not show this? Okay? Now, I guess we're very happy that this is not a 500 or 600 level organic chemistry class or biochemistry class, so we really only have to focus on a couple of small things here. Okay? One, you already know that all these steroid hormones are all derived from cholesterol, top left. Okay? And basically what happens, all these red arrows are all kinds of different enzymes that will take that cholesterol, turn it into pregnenolone, progesterone, and then we get either aldosterone or cortisol, testosterone, et cetera, et cetera. Okay? So the most important part initially in reproductive physiology is taking cholesterol and turning it into progesterone. Okay? Bless you. From progesterone then, we go all the way to making testosterone. Okay? Testosterone is a precursor to one hormone we already talked about, dihydrotestosterone. Okay? That is the 5-alpha reductase. Testosterone is also a precursor molecule to another hormone, estradiol. And estradiol, from now on, the few times I put it on the board, that means, or E2 means estrogen. Okay? Estradiol. Okay? So testosterone gets converted into estrogen, and this is due to a hormone called aromatase. Okay? So, very quickly, first question. Do men have progesterone? Do men secrete progesterone into blood? No. Okay, so we were almost on the right track. No, if females do, because what we'll talk about next week is progesterone doing a whole bunch of things. We're really not going to talk about progesterone at all in the male, except it is a precursor. We need to make that first before we can make testosterone. Okay? Now, everybody in the room here have testosterone? Yep, everybody does, right? Do all the men in this room have a lot of aromatase? No. Okay? No. That would mean that you're making a lot of estrogen. Okay? And generally speaking, blood levels of estrogen in men are low, testosterone levels are high. Okay? That's why we call them sex hormones, which is kind of stupid, or male or female hormones. Okay, so does that kind of make sense? That's all I want you to get from this graph. That starts with cholesterol. Progesterone is a precursor to testosterone. Testosterone is also a precursor to dihydrotestosterone and to estrogen. Okay? And no, aromatase takes testosterone to E2, and 5-alpha reductase is necessary for DHT. Okay, sex steroid hormones. We have three kind of broad classifications. We have androgens, estrogens, and progesterone. Progesterone is just a precursor. Androgens are these two hormones we just talked about, testosterone, dihydrotestosterone. And estrogens are estradiol and all that stuff. Okay? And I hope that we still remember from Dr. Warren's lectures that steroid hormones are lipophilic, bind to intercellular receptors, and it leads to transcription. Okay, so usually the effects of a steroid hormone, it takes a little bit of time. Okay, it does lead to changes, but it's going to be a little slower than, for example, a peptide hormone or something else. Okay, also means that steroids, the way it travels through blood, is bound to proteins. Okay, those steroids are not just dumped into the bloodstream. Okay, that's quickly the endocrine stuff. Don't worry, the endocrine stuff will come back. I know how much you guys love it, so. But let's start briefly. Again, I hope that this is somewhat familiar to everybody here, so I guess a quick review. What we'll do is take a quick tour through the male reproductive tract, okay? So I think there's about maybe 60 students here, so let's pretend that this classroom is a testicle and there's 60 sperm inside, okay? And now we're just gonna go out. So we start here in the testis. The first structure that we see is the epididymis. Okay? The epididymis is nothing but a highly convoluted single tube. Okay? There's the head of the epididymis, that's the top, that's where sperm enter. Then they go through the body of the epididymis, the tail of the epididymis, and then the epididymis kind of straightens out and it turns into the vas deferens. The vas deferens goes into the body, all the way around the bladder. And then what happens is it starts fusing with a variety of ducts coming from accessory glands. Okay? I know that you guys are all experts not only in about the kidney, but also the GI tract. So think about it as the liver and the pancreas also added lots of stuff to the GI tract. That's the same thing here. We have accessory glands like the seminal vesicle, the prostate gland, and the bulbourethral gland that all secrete fluids and buffers and nutrients, glucose, fructose, that kind of stuff, that all goes to the lumen of that tube. Okay? Once we have all these tubes, the vas deferens and the ducts from the seminal vesicle, the prostate glands, and the bulbourethral glands all fused, now we have a urethra, and then out through the penis. Okay? So what's labeled in blue are the accessory glands. What's labeled in red, except for the testis, that's really the duct system. Okay? Now, I hope we still remember what the Wolffian duct does, right? Wolffian duct, epididymis, vas deferens, seminal vesicle. So in fact, two parts of the duct and one accessory gland. Okay. Okay. If we would take a peek inside the testis, this is what we would find. Okay. If you remember from yesterday, we talked about a fetal testis, this whole hot dog on the bun thing, that the testis kind of looked like this, and you had all these cords, right? These we call testicular cords. Do we remember what was inside the testicular cords? What's the two cell types? Gotta love all the repetition in here, right? So testis, right? Sertoli cells? Theca cells are not in the testis. That's all female. Germ cells. Sertoli cells and germ cells. Okay? Sertoli cells and germ cells are in the testicular cords. Now, this was all during fetal life, in a fetal testis. We skip birth all the way to puberty, and now we're in adult testis. And we don't call it testicular cords anymore, we call it seminiferous tubules. Okay? That's the only difference. We just give it a different name. In terms of cells that are present, it's the same stuff. Sertoli cells and germ cells, that's it. In between the seminiferous tubules, we have interstitial space. Okay? So interstitial space, that's where we have Leydig cells and blood vessels. Okay? So when Leydig cells secrete testosterone that goes into the bloodstream, and now it can go all over the place. Okay? All these seminiferous tubules that are here are highly convoluted. They all come together in smaller network, a smaller number of tubes, and all eventually in a single tube. That's the epididymis. Okay? And here it kind of shows this is where sperm first enter the epididymis, the head, then the body, then the tail, and then the tube kind of stretches out, okay, and becomes elongated. Okay, we already talked about the fact that the testis is weird, well, for many reasons, but it's outside of our body, okay? And this is important because testis function in terms of making sperm requires a lower temperature. So instead of 37 degrees Celsius, it is about 34 to 35 degrees Celsius. So the two ways that you can do this then, well, obviously one is testicular descent, right? So basically testicular descent is we're going to put it outside of our body. And the other one is something called the papilliform plexus. The papilliform plexus is a countercurrent exchange between arteries and veins. Okay, the papilliform plexus. So this slide, which is the same as the previous one, actually highlights the papilliform plexus right here, the countercurrent exchange. Okay, so the way this helps with cooling is that you have testicular arteries coming from the body going into the testis, okay, carry warm blood. But the first thing they see is testicular veins returning that carry cold blood. So the warm blood is already being cooled by colder blood. Okay, and that is the papilliform plexus, and that's right up here. Okay. Okay, then the next thing is testicular descent. How the heck do we get these testes outside? When do you think this happened? When does testicular descent happen? In utero or puberty? In utero. Okay, if your testes have not descended by the time you hit puberty, there's an issue. Okay, they should have descended, be descended either when you're born or at least after the first year of age. Okay, so this, what we're talking about right now is again something that happens in utero during fetal life. And because you guys always appreciate my drawings, we'll do another one. So this is kind of a side view of the body cavity. Okay, and what we're going to do now is kind of talk about the first stage of testicular descent, which is abdominal translocation. Abdominal translocation just really kind of means movement of the gonad through the abdominal cavity. Okay. Now, when we talked about the fetal gonads, ovary and testis, doesn't matter, they actually form right underneath the kidneys. So the adrenal glands were on top, the gonads were right below the kidneys. Okay. And in both cases, that gonad is kind of stuck to the body wall by a ligament. So this is the case for both ovaries and testes. Okay? And this ligament is called the gubernaculum. Okay? And so what happens now is, at some point when I have a testis forming, the testis secretes testosterone, and it also starts secreting a hormone called INSL3, another hormone. This INSL3 coming from the fetal Leydig cells, okay, acts on receptors that are on the gubernaculum. And what it does to the gubernaculum, that ligament, it makes it really thick and strong, okay? So that gubernaculum, that ligament becomes really thick and really holds onto that gonad very tightly. Now what happens at this point is during fetal development, hopefully it's not too much of a shock, but the fetus grows. Trust me, it does, okay? So when the fetus grows, I want you to start kind of thinking, well, look at what happens to this gonad now. I mean, basically it is held in place right here, and the rest of the body kind of grows upward. Okay. Does it kind of make sense then that how that gonad just kind of, well, it passively just stays close to the body wall near the bottom? Okay, the rest just kind of grows away from it. Now the ovary, on the other hand, that ligament is kind of thin and not really as strong, so it doesn't really hold that ovary in place, and the ovary kind of, you know, stays up higher as well. Okay, so the gubernaculum holds that testis in place. So that is abdominal translocation. Okay, that's the first part. The second part is something called transinguinal migration. All this means is that we have a hole or a tunnel in our body wall that is called the inguinal canal. Okay? So transinguinal migration just means that the testis moves through that little tunnel, the inguinal canal, through our body wall. Okay? And then finally, inguinal scrotal migration, all that means is now it's near the end of the tunnel, and now it moves all the way into the scrotum. And that last part is dependent on testosterone. Okay? So that's really dependent on testosterone. So this is very important. This is an important process because if this does not happen, like I said, testis function cannot occur. What's even worse is if this never gets corrected, then if that testis stays inside the body cavity, it can actually become cancerous. And actually, people that have an undescended testes, if that does not get corrected, have a much greater likelihood to developing testicular cancer. Okay? The other thing that is of great interest to a lot of people is the observation that in many, many Western countries, the incidence of babies born with undescended testes has increased dramatically to a point that in certain countries, it is almost 1 in 10 baby boys that are born, or 10%, have undescended testes. Okay? So like I mentioned before, it's not like right at the time of birth it would be surgically corrected. If this doesn't fix itself, then definitely by the time they're anywhere between 6 months, 9 months, a year old, then they'll go in and quickly do that. And again, it's important because if that does not get corrected, higher incidence of testicular cancer, and on top of that, you'd definitely be infertile. Spermatogenesis cannot happen, and basically all the germ cells will be destroyed if it stays at 37 degrees Celsius. Okay? Any questions about this? Okay, that's enough testicles for today, and have a great weekend. I'll see you Monday morning.